Hi, and welcome. This is Peter Deep with Lambda. Thank you for joining the webinar today, Assessment of the Humoral Response to SARS-CoV-2 Infection via Novel Multiplexed Assays. It is now my pleasure to turn today's program over to our presenters, starting with Dr. Gebel. Dr. Gebel?
Well, thank you very much, and it's a pleasure to be here this afternoon. I want to say good morning, good afternoon, or good evening, depending on where you are in the world. It sure is a different world than it was last year at this time. By the middle of March of this year, the world was fully engulfed in the COVID-19 pandemic. And around that time, there became a clamoring for not testing to determine who was infected, but for testing to determine if somebody had been infected and had they made antibodies. And with that, the FDA decided on the 16th of March to issue a policy with somewhat relaxed regulatory flexibility, leading to more rapid testing capacity within the US. And the hope was that there would be many individuals, many different companies that would provide testing, allowing for expansion throughout the United States and a lot of testing to be performed. And with that, the floodgates opened. And in fact, there were 170 different tests that became available. And not all tests were the same. More importantly, it's important to know that out of those 170 tests that had been developed, only 158 of them had not had any review whatsoever by the FDA. Instead, what the FDA allowed in this relaxed flexibility was for the vendors and manufacturers to provide that data without an FDA review so that the tests could be distributed. At that time, shortly thereafter, it became clear that not all of these tests performed the same. Somewhat were not working at all, and in fact turned out to be fraudulent. On May 4th of this year, the FDA changed their rules and required that any tests that was on the market, there had to be submission of data to the FDA for them to review in order for them to get FDA approval in terms of saying that it had reviewed at least some of that data. But with that in mind, let's just say that all the tests were working, and if they were, what kind of tests were available? There were two major types. There was the one on the left, which is a point of care testing. That's referred to as a lateral flow assay. It should be very reminiscent of what you see in your pharmacy when you want to have a pregnancy test performed. It's the same basic principle. On the right side of the screen is the other type of assay, which is a form either of an ELISA assay, a chemoluminescence assay, electrochemoluminescence, or fluorescence. But in both situations, the platforms are using COVID-19 specific antigens. But the antigens that are on these platforms aren't necessarily the same from test to test. And that's a very important caveat. Furthermore, the tests that are performed typically at this time, or back in this time, looked at one antigen at a time. So let's start with these point of care tests where I have some personal familiarity and to give you an idea of what we were dealing with at this time. In fact, Bob shared this data with me, and that was my first exposure to this lateral flow. And what he presented to me was 20 different lateral flow results. And of the results, what you were looking for was a band that was positive. This is a positive band. This is a control band showing you that the assay worked. So what you're looking for is a positive response with that band. Among the 20 samples that I saw, 14 of them were clearly positive. They had a nice strong band. 3 of them were clearly negative. There was no band there whatsoever. And the last 3 were ambiguous, where everybody on this call would look at each other and ask, well, what do you think, positive or negative? So that in itself wasn't so bad until I found out that all 20 results came from the same serum sample run by the same testing technologist all at the same time to eliminate any variability. That is incredibly problematic. And so one has to, even today, with the tests that are currently out on the market right this moment, those tests, they still have false positive and false negative results. The specificity and sensitivity is approaching what we need it to be, but clearly at this time it wasn't anywhere close to what was necessary. And again, as I did say, the antigens that were being utilized weren't the same from test to test. And even if you have a platform where the requirements are the same for an individual, not again were all samples going to be the same. For example, if blood was required, somebody might require a finger stick and somebody else would take blood from a vein. And samples might be serum samples, they might be plasma samples. All of these contribute to variability that you have to be aware of. But let's make this a given for a moment and say that all the tests worked as best as they possibly could. Under those circumstances, what are you actually getting? And the answer is for the information is simply a yes or a no. That's all we really got out of the tests that were available at that time. So more importantly, I believe the question is, what information don't we have? And the information that all of us would like to know is if the antibody is there, is what, how strong is it? What's the titer? How stable is it? Is it going to last a week, a month, 6 months, a year, 10 years, a lifetime? We had no idea. We still don't have an idea of all of those answers. There's a lot of data in the literature now suggesting that looking at subclass specificities for IgG and maybe even for IgA could help in elucidating some of the function of these antibodies. For example, whether they're FC receptors, whether they bind to FC receptors that trigger macrophage-mediated cytotoxicity or antibody-mediated cellular cytotoxicity. We don't have the answers to that yet. We also don't know the functional properties and fine specificity of these antibodies. And by that, I mean, we don't know whether they fix complement, and more importantly, we don't know for sure whether they're neutralizing. There are some studies that have been performed back in March, April, and early May in vitro that showed that at least some of those antibodies did have neutralizing capability, but they were all done within a test tube. We didn't know whether those antibodies had any neutralizing activity in vivo, so we had no idea whether they were protective. And that becomes one of the critical components of any assays that are available. You're making antibodies, but are these antibodies going to be protective? So what else don't we know? Well, there's quite a bit that we don't know. So for example, as I said, the two different platforms that were available tended to look at one of two antigens. They either looked at an internal protein, a nucleocapsid protein, or they looked at a spike protein. And these are all these little red spikes on the surface of the coronavirus. And in particular, the majority of assays that looked at the spike protein looked at a particular region of the spike protein called the receptor binding domain. And the reason that the interest was in the receptor binding domain is this portion of the spike protein that actually binds to its receptor on cells and tissues, the ACE receptor. And so it made sense that if you had antibodies to the RBD, that you would be able to at least predict that those antibodies had neutralizing activity that could prevent infection. And it certainly is a reasonable idea. There's more data in the literature now that you'll be hearing about a bit later, but it was a good start. However, it struck me right away that looking at only two proteins and antibodies to two proteins doesn't tell the whole story. In fact, the SARS-CoV-2 genome has a total of 29 different proteins. And the open reading frame proteins, there are actually 16 non-structural proteins encoded there that have all different kinds of activity, ranging from sabotaging the cell from making any of its own proteins, to inhibiting interferon production, to making sure that the viral machinery is present to make as many copies as possible. There are also accessory proteins that are encoded, and there's data that was out even as early as April that indicated that antibodies against at least some of these non-structural proteins and accessory proteins were developed in individuals who had been infected with SARS-CoV-2. Whether they had any functional activity, whether they could be protective, is unknown. One might think that because they're internal proteins, that there was no way that an antibody could have any protective effects. That's not necessarily true. There are viral models like dengue hemorrhagic fever where antibodies can actually exacerbate the disease. They're called enhancing antibodies. And there is now some data coming out in the SARS literature, at least in vitro and in monkey models, that some antibodies that are made may in fact exacerbate the disease. They might even be involved in some of the syndromes we see among children who develop a multi-inflammatory like complex syndrome that may be related to the antibodies. There's so many different questions that could be asked, but we didn't have enough tools. It struck me right at this time that what we were missing was an ability to look at multiple proteins at the same time. Now, it just so happens that about 2 weeks before I started really getting into this literature, I was at a meeting in Arizona. It was a transplant meeting, and I had the good fortune to meet Nicole Brockway, who, as you heard, is the president of transplant diagnostics at Thermo Fisher. And as I was thinking about this, that a really good test would be a multiplex assay, I decided to give Nicole a call and find out whether there might be any interest on her end. And not only did she respond enthusiastically, she had the vision of what a test like this could do for the community, and she absolutely signed on to the idea. At that point, I reached out to some of the smartest people I knew to work on this project. Unfortunately, none of them were available, so what you're going to be hearing from are the people who are on the rest of this call and who did the work. Of course, I am kidding. The people who are on this project are some of the smartest people I've ever worked with. They are incredibly intelligent, hardworking, passionate. This project has been productive. It's been a lot of fun, and I look forward to working with each and every one of my collaborators over the next several months and years. With this, I will just about turn it over to Bob. We started with the idea that there was a theory that looking at these antibodies was going to be useful. And in theory, there's no difference between theory and practice. In practice, there certainly is.